Thanks a lot. I wanted to keep that suspense for a little bit later. So now everybody knows already what will be the talk about today. So today will also be much more a tutorial than a lecture, right? So I want to guide through you step by step how to use these machine learning methods, okay? But I want to use that for the second part of this talk. Also, I was asked to give a bit of explanation about this FLEX mission, which is where I'm currently working on. Again, I replace here Professor Jose Moreno. Normally, he was supposed to talk about that. So just 5 slides about the FLEX mission, and then afterwards, we move towards machine learning. Okay, so let's have a look to our planet again, our living planet that provides food and feed to all of us. But this time, the question is rather how not just to quantify vegetation properties, what we saw yesterday, but now let's try to go up a little bit more in depth, be more ambitious, and try to quantify vegetation productivity. How is that typically done? Traditionally, we have our optical Earth observers, Sentinel-3, Sentinel-2, and so on. So the thing is that they measure reflected radiation, what we saw yesterday. However, with reflectance, there's a few main problems. Most of all, when we are interested in vegetation, a pixel never contains a vegetation only. It's always contaminated by bare soil, man-made surfaces, and so on. So when we try to retrieve information from reflectance, we are never sure, are we really watching at vegetation only, or has it also contamination of other kind of surfaces? So that's the first problem with reflectance. Second, maybe most of all, we are able to derive some vegetation properties. You can think about chlorophyll content and so on. But what does that actually say about vegetation productivity? Chlorophyll says something about the status of the plant, but it doesn't say anything if the plant is under stress, is it healthy, or so on. You can have a perfectly green plant, but when it has water deficiency, it will be under stress, and actually it will no longer be producing. So reflectance has several limitations when we want to quantify vegetation productivity. So that's why that ESA approved to move towards a new mission. It's called the Fluorescence Explorer. FLEX mission. It will be launched by 2022. And that mission is dedicated towards vegetation observing, but not just by means of reflectance. It also observes reflectance, but actually it searches for a signal that comes from the plant itself, namely chlorophyll-induced fluorescence. That signal comes directly from the photosynthetic machinery. So when the plant is photosynthesizing, some of the energy is released, and that is what we try to measure. So in this way, we have a direct link towards photosynthesis, okay? So what is this fluorescence? It's the glowing of the plant when photosynthesizing. We are not able to see this in the naked eye, but in the lab, with the right filter, it's very easy to see how plants are glowing. Everything that is photosynthesizing emits fluorescence. So it's a signal we can always capture, and we know the plant is still alive. What are the mechanisms behind? So yesterday we already saw that most of the light is absorbed by the plant, by the chlorophyll, the chloroplasts. Most of that light actually is too much light for the plant itself and is being dissipated by all kinds of mechanisms. Eventually, it's converted into towards heat. A small part of the energy that is being used for photosynthesis escapes the plant as fluorescence. This part that is actually escaped by means of the photosystems, which do the conversions towards photosynthesizing. And there are 2 photosystems, photosystem 2 and photosystem 1. When they are releasing energy, you can see they provide these 2 peaks. Oh, okay, so here and here. Okay. So just to be clear, fluorescence is not one signal, it's not one wavelength. Actually, it is a broadband signal from 650 until 800 nanometer. And these peaks are highly dynamic, depending on if the plant is under stress, how is it photosynthesizing, what kind of plant, say trees or 4. These peaks, they are changing. So if we are able to quantify these peaks, we are able to know how healthy the plant is, what kind of condition it is it's actually living. Of course, the situation is a little bit more complicated because this is not only a very small signal, but also it travels through the leaf where there's scattering occurring. It travels through the canopy where you also have this scattering and reabsorption by other leaves occurring. And eventually, it scatters through the atmosphere. So all these parts need to be known, preferably quantified, in order to be able to detect what is going on under here. So basically, it's a scaling problem. Other pigments, there are these energy being absorbed and being used for photosynthesis. that is actually part of the leaf is part of the canopy is part of the landscape, and when we try to observe it with satellites, we need to be able to know the whole path, what is happening. So this brings us to the FLEX mission. FLEX will fly in tandem with Sentinel-3 because a lot of additional information is actually required in order to be able to interpret the fluorescence signal. For instance, temperature. Temperature also determines a lot how well the plant is behaving and how much it is photosynthesizing and thus fluorescence emission. Sentinel-3 provides also temperature, so we make use of Sentinel-3 in order to better understand the fluorescence signal. The main objective of FLEX is to quantify actual photosynthetic activity of terrestrial ecosystems from Spain, so accounting for vegetation health status and stress conditions. So basically, it's this upscaling problem, and there's a lot of research already being done at the leaf level by plant physiologists. These are the products that we aim to retrieve. So we aim to retrieve first in the oxygen absorption bands to retrieve the fluorescence. Based on that, we try to retrieve the full fluorescence spectrum. From this, we can derive the total fluorescence, so the total fluorescence yield. And that afterwards, we try to decompose into this PC2, photosystem 2, photosystem 1, to be able to know under what kind of stress the plant is. There are all kinds of campaigns going on currently, and we have already quite good understanding on mapping these fluorescence at the airborne scale. However, when moving upwards towards space, it becomes more complicated. Again, fluorescence depends on the plant type. It depends on time of the day, time of the year. If the plant is under stress, so it's highly dynamic, which means that it's very hard to correlate it directly towards photosynthesis, GPP or so, but actually much more information is needed in order to properly interpret the signal. So that's why FLEX will fly in tandem with Sentinel-3, and this is a truly synergistic mission. We do synergy for atmospheric correction. We do synergy, we make use of both data types for these plant vegetation properties retrieval, LAI, chlorophyll, and so on. And at the end, all this data is being put together into a simulation system, into a photosynthesis model in order to derive GPP, for instance. So you can imagine we don't go immediately from chlorophyll fluorescence towards GPP, but actually we go for a more mechanistical point of view to see how all the energy is flowing. So for this, we need to know about the temperature, we need to know about the optical properties, the fluorescence, the pass-through retrieval, so that we have the vegetation properties, we have the full fluorescence peaks. At the end, it's all being put into a photosynthesis model, and from there we can derive interesting products such as GPP. So this about FLEX. Now let's go to the results of the voting yesterday. Well, it was already been mentioned. There's one clear winner, which is this machine learning, and to my surprise, the second most voted were the vegetation indices. So I will spend a few words on vegetation indices later on today, but the tutorial for today will be about machine learning, and then maybe tomorrow we will have a closer look about these RTMs, how to use them, and we can combine that with some applications of the RTM. Okay, this we've already seen, so that's the basic idea of what we will do today. Now it will be more a tutorial. I will not do the work. The computer will do the work for me. I prepared a few small exercises. We go step by step through it so that you all can see it's quite easy to use them, and it has all kinds of applications. Also, I want to emphasize that the data I'm using is optical data, and it's for vegetation properties mapping. However, at the end, what we work with is just rows and columns. So it can be any kind of data. If you are more into ocean or atmosphere or so, or even radar, at the end, what we need is the data where we try to derive our properties from, so spectral data or radar data, and our variables, in my case, vegetation properties, okay? So the type of machine learning algorithms, we've seen this yesterday also. Basically, they all come not from myself, but from the group who's just sitting next door, the signal processing group. And it also does not come from them, but they collected all the codes they could find in the MATLAB environment, and by now we have more than 22 machine learning algorithms putting together in the library. So it means that you can just select one, compare against another one or so. The source code is provided here, and basically what I provide is just a very user-friendly graphical user interface around it. Maybe one algorithm I would like to spend a few more words. This is Gaussian processes, and I'm very fond of that because it's a Bayesian algorithm, so it provides probabilistic outputs, which means that it does not provide just an estimate, but also uncertainties around it. And another nice property is that it needs much less sampling data than, so to say, neural networks or the other classical machine learning methods. Yesterday, there was a question about how many samples are needed. It all depends a lot about the kind of algorithm you're using. With Gaussian processes, with very few data, you get also excellent results. And not only that, it also provides you insight in what are the relevant bands. So it immediately gives you kind of a sensitivity. What are well, for optical data, the most important spectral domains that are playing a role in the regression model? So it's not really black box, but you have insight in where the information comes from. And also it provides these uncertainties. And these uncertainties are quite handy because when we apply it for mapping, so we do a little bit of training, we validate it, and we apply it to an image. So we have our map, in this case chlorophyll content, and we also have the uncertainty map. And this uncertainty map, what does it say? Basically, so the lower, the more certain it is about the estimates. So for instance, this part here is retrieved with high certainty. However, there's this small region here that provides high uncertainties. So what does it say? So it does the estimates, and you can see the estimates look exactly the same as for the other parts of the parcel. But basically, it say, I'm doing the estimates, but I'm not quite sure with my model if I'm doing a good job. And why would that be the case? When we go back to the original image, it appears that it's a bit darker. Actually, it has just been irrigated. So the soil is darker, so the signal reflectance is slightly different. It still does estimation, I would say very good. It just say, hey, be careful here. I'm not sure if I'm doing a good job. And that's very handy because these estimates, these uncertainties, they allow us then to go back into the field maybe and to check what is going on, or maybe get some more data, put it also in the training so that we are also certain for this kind of land covers. Okay, so always I would say uncertainty is crucial when we try to interpret a map. So this brings me to the software that we have been developing for the last 10 years. It's freely downloadable. The only constraint, it's in MATLAB, so you need to have a MATLAB license. You may have also you need the machine learning toolbox within MATLAB. But further, yeah, you can just download it and use it for your own purposes. Today, I want to focus on the machine learning toolbox, but you can see it has all these relative transfer models, and the idea is that the outputs of these relative transfer models can then be put into the machine learning or the vegetation indices or the inversion toolboxes, so that you can actually, you don't really need to have field data, basically. Yeah? You can train with simulated data. Maybe field data is needed for validation, but you can develop your own models just by means of simulated data. Okay, so the next couple of slides, I will quickly go through the steps we will do it afterwards. So just to make you familiar with the steps that are required. Then afterwards, I will go to MATLAB and try it myself, and just fingers crossed, nothing's go wrong. I tried it yesterday evening, but always once you are standing on the stage, the pressure is high. So I apologize already if something would go wrong, but let's just hope the best. Okay, so the data flow. Basically, what we start with is, in my case, spectral data and variables. So we have maybe Sentinel-2 data, and we have some field measurements of chlorophyll, LAI, or so on. And it goes through, that's the input. We will configure the settings, so that we select the type of machine learning algorithms we are interested in. We do the validation. If we are happy with it, we apply to an image, so retrieval. This can be broken down into the input data we will keep a part apart for training, and the other part we will use for validation. So this is the training part, validation part. In case we use simulated data, what we typically do is we add a little bit of noise, because simulated data is so smooth that it's actually too perfect for applying it towards a real image. That's always a little bit of noisy. If we use real data, what we will do now, there's no need to apply additional noise, because the real spectral data is just as noisy as the image, if it comes from the image. So we apply the regression algorithms. We, against validation, we test the algorithms. We apply all kind of goodness of fit functions, R-square, root mean square error, and so on. If we are happy with one model, we apply it, and we have our map. In Artmo, what does it mean? It means a few steps. So first we have our input data. We load it. We select our machine learning algorithms. We select a few options, how much data is for training, how much is for validation. It does the validation of all the selected algorithms. You can plot some goodness of fit statistics. And afterwards, you apply towards an image, and you have your map. So for today, I want to show you just a few options. So we will only make use of what we call user data, so data that has been measured in the field or spectral data that comes from an image, and not simulated data, just because I want to be aware that it does not necessarily have to be optical data or for vegetation, but user data can be any type of data, okay? We will show single output models, so you make a model for one variable, LAI, chlorophyll, and also multi-output models. Multi-output models, those models, they produce output of a range of variables. So, it can be 3, 5, just as many variables that you put into it. And then we apply mapping. So the input data, how we like it is that we have a few rows of variables. In this case, this is chlorophyll content, LAI, fractional cover, biomass, and so on. And then below, we have the spectral samples. So here we first have the wavelengths. And in this case, these are just the measurements we're taking in the field. With the GPS, we have the coordinates, and these are just the spectra of the pixels. In this case, it was from a CRIS image, so this was a hyperspectral image back from the 2000s or so. But it can be airborne, or nowadays it can be a Sentinel-2 or so. So that's all what you need to prepare, just this file with your variables on one hand and your spectral data on the other hand. Yeah, maybe this is a little bit too much information, but basically later we will see you load it. It checks if all the data, all the columns, the cells are completely filled, if there are no weird values such as characters or a none in there. And then basically you simply have to say the first row are the samples. The second row is chlorophyll content. So you just say leaf chlorophyll content is line 2, and you add that. So in this way you add the variables where you want to develop your algorithms with. And then the only more information you need to give is from where the spectral data starts. So in this case, it starts from row 5, and that's all. So then the next step is the selection of the machine learning algorithms. By now, we have about 22, and you can just select as much as you want of them, and then it will run them step by step, one by one, over them. You have the option to add noise. If in case you use simulated data, it's recommended. And you simply have to say how much data you want to use for training and how much is then used for testing. Further, there are all kinds of other options, such as cross-validation. In case you have very little data, maybe you want to apply cross-validation methods that all the data is used for training and validation. We also have some band tools, such as dimensionality reduction. In case you have hyperspectral data, maybe you first want to apply principal component analysis and then do the regression over the components. And then essentially, once you made your selection, you click on finished. And then the final step, okay, so we have the same for the multi-output algorithms. We have 5, but it's exactly the same. So here you can develop models for immediately multiple variables. And then here are these dimensionality reduction options. So principal component analysis is the most known, but there are many more of them. In fact, here we have put together 11 dimensionality reduction methods. So also you can select more of them, and you can see which one is better performing. Interestingly enough, PCA is not always the best performing one, so we can still improve our algorithms a lot by taking the right dimensionality reduction methods. Also, you can play with the number of components. Typically, when you add more components, more information is added. So also there you can improve a little bit. But here it's all automated, so you can just select all these ones. It will do the analysis all over them, and at the end, it gives you the best performing results. And here is some words about the cross-validation option. So we have k-fold, for instance, or leave one out. So basically, it makes subsets. It trains using the subsets, leaving one subset for validation. And at the end, it moves over all the subsets so that all the data at the end is used for validation. So finally, you give your project or exercise a name. It calculates all kinds of statistics, and then it stores them within a MySQL database which runs underneath Artmo, so you can always go back to your results, and afterwards you just retrieve the results you are interested in. So the the final table will give you an overview of your algorithms and then all the statistics, so the R-square, root mean square error, and so on. And it sorts according to the statistic you are interested in. So maybe you want to know best performing according to root mean square error, or normalized root mean square, R-square, or so on. So it gives you this overview. And then further, there are all kinds of options for plotting the output, estimated against measured, the residuals. For a few discussion processes, it shows also what are the relevant bands. And eventually, you just select one, perhaps the best performing one. You can select actually multiples. And these ones are then being used for mapping applications. So then a final step, here is a selected algorithm chosen. But if you are not willing to do this validation, you could immediately go to this GUI and just select an algorithm, say, I use all the training data for training, and it afterwards immediately runs against an image. So basically, we have validated this algorithm. And now by clicking on OK, we simply select our input image, and we select the name for the output map, and that's all. Then afterwards, our map is being produced. Then we have some options also to visualize it nicer, to change the colors and so on. So let's go to the exercise, and I will focus on 3 variables, so leaf chlorophyll content, leaf area index, and fractional vegetation cover. So we use a CRIS satellite, which looks like this. It's a kind of hyperspectral image. It has 62 bands, and the pixel size is up to, if I remember, well, 34 meters. So basically, you could compare it like a simpler or smaller version of Sentinel-2. First, I wanted to show Sentinel-2, but these images are so huge, it will slow down now this lecture. But if we have time enough afterwards, I can show the same for a Sentinel-2 image. Also, if we are interested more in hyperspectral data, I could also show a high map image, so which has 125 bands. And I want to show that because the more bands it has, the more complex it makes for the machine learning method. And when you have many bands, it's really recommendable to first apply these dimensionality reduction methods, these PCA. You reduce your bands into some components, and then you apply the machine learning algorithm. OK, so we will go through these steps. And these are the small exercises I prepared. So we will do first for CRIS, and then maybe some Sentinel-2. And now I will switch towards MATLAB, and let's hope the best that it will work out. So let's have a look. Everybody can see, maybe it's a little bit small, but so the first thing I will do is just I type Artmo. And now it will load all the relative transfer models and check the MySQL connection, and then load the apps, such as retrieval toolboxes. And then it should open. OK, so for very new users, the very first thing to do is to create a new database so that you have just an empty space where all the results will be stored. So I will quickly create a new database. And now the spaces are being created where we can store our results. So this has to be done only just one time when you download this Artmo toolbox. OK. Now we go to retrieval, we go to the machine learning toolbox. So we have to go through the following steps. First, we do input, then settings will get activated, we validate, and we apply to an image. So first input, we have the option for simulated data or user data. So now I go to user data, and here appears this window for loading a text file. So I go to browser, and I prepared CRIS data. So here I have this text file. I guess it's this one. OK. No. Oh, sorry, it's this one. So first, it shows a piece of the data to see if it's the right data. If maybe you want to remove a header line, if it's the right data, you click on OK. And now MATLAB checks if is it readable as a matrix. So I'll find, and now we simply have to say which line is which variable. So I know that the second line is leaf chlorophyll content, line 2, and I say add. I know that the third line is leaf area index, and I add. And then further, the following line is fractional vegetation cover, line 4, I add. And I say that the spectral data starts from line 7. So that's how you could save these settings that next time you don't have to do these steps. But now I will just simply import. And now I can go to the second step, which is the selection of the machine learning algorithms. So settings, I can choose either single output or multi-output. So let's first start with single output. And here, there is this overview of all the implemented algorithms. So as a reference, we also added just linear regression, which is like the basis. And then you can see how much better your machine learning algorithm is doing as compared to classical linear regression. So we have the principal component regression, partial least square regression, and then there are all kinds of random forests. Then there are all these kernel-based support vector regression, kernel-rich regression, neural networks. For neural networks, by the way, there are some advanced options that you can define the design of your neural network also. All the others, they have just default settings. So let's select a few of these algorithms. I will not do all of them. It will take too much time, but just for you to get an idea. So let's select linear regression as a reference, maybe also principal component regression, partially square regression. These are widely used. I also select kernel-rich regression because it's powerful and fast. And then finally, also Gaussian processes regression. And let's say 50% of the data we will use for training, so the other 50 we will use for validation. Later on the results, we will only show validation results, because with machine learning, the training is always extremely good, way too good, always an R-square of 0.99 almost, because of this tendency of overfitting. So it's not training we are interested in, it's validation we are interested in, okay? And so Chris has about 62 bands. I could apply dimensionality reduction, but to start with, we just try all the bands. And let's see how it goes. So I click on done. And now validation, I just give a new name. So this exercise, let's call it Chris. We have 3 variables. We did machine learning. I just use a name for that. Later on, I can easily find back what was in there. And we use 50-50 training testing. OK, so now it will start running. And you can see MATLAB. So first it does this linear regression, and then PCR, partially square regression, kernel-rich regression. And it will do that for the 3 variables. We did chlorophyll, LEI, and fractional vegetation cover. And then the results appear. So we can have a look. Now they are sorted according to normalized root mean square error. To be sure, let's sort them according to R square. OK. And so we can see the best performing one is on top with very nice results. In this case, for leaf chlorophyll content is kernel-rich regression. And then the second one is Gaussian process regression. So these are the results for leaf chlorophyll content. But we can also show them for leaf area index and then for fractional vegetation cover. So let's have a look maybe for leaf area index. And if we click on graphics, we have all these options to have a closer look to the results. So if I click on plot, measured versus estimated. So these are it's a one-to-one line for the validation data, right? And we see that they are very nicely follow the one-to-one line. Another option is the residuals. So we can see how much it deviates from 0. And if I go select these Gaussian processes, there's another option for the sigma bands. And so this is the sensitivity of what are the most important bands in the regression model. And OK. So the lower the sigma, the more important that band is. So we can see that some bands here at 520 so indivisible, but also some bands maybe in the red edge they are the ones that drive this regression method, okay? So we can do that for LAI, and we can do exactly the same for kernel, for leaf chlorophyll content. So this is kind of a sensitivity study that immediately you can see what are the bands that are of importance. So you could actually decide, you get rid of these bands that does not provide any information, but you only select those bands, and then you can even come up with a more powerful regression method, okay? So after your inspection of your results, let's say that you want to use one of those for mapping, okay? So then you select one of these methods. I want to select Gaussian processes, for instance. You can select multiple ones, so but now to keep it simple, let's just do only chlorophyll content mapping, and we are done. So we can go to the next step now, which is the mapping part. So we go to retrieval. You can do either apply it to an image or just to spectroscopic data. If you maybe have field measurements, you can also convert it then into estimates of variables. But now we want to process an image. Here is our selected and validated method. And all we have to do is now to select an input image. So let's select this one here. And the output, so the output, it gives me Gaussian processes, leaf chlorophyll content, and now it starts running. So this takes maybe a little time. Okay, here it is. And now we can see to the output maps. So with Gaussian processes, we have our estimates. Also, we have these uncertainties. We have what we call absolute uncertainties. So these are the standard deviation around the estimates. And relative uncertainties, which is the coefficient of variation, so the standard deviation divided by the estimates and expressed as percentage. So let's first have a look to the leaf chlorophyll content map. So this we can put here. This looks very nice. This is taken over a Spanish region, Barracks, so in the middle of Spain, more or less. Has somebody any idea what are these circles? So we can see high values over circles and further very low values over the remaining part of the image. Someone of Spain probably can immediately say that these are irrigated areas. So in Spain, it's super dry. In summertime, there's no more green vegetation, only those that are being irrigated. There we still have high leaf chlorophyll content. So this is the first part of the information, but let's have a look to the uncertainties, because maybe that's even more interesting. So first, let's have a look to the absolute uncertainties. And we can immediately see that it's all very low everywhere. However, there are a few areas that provide high uncertainties. Now, what does this information tell us? Basically, it says that here we are uncertain of these areas, but why are we uncertain of these areas? Has anybody an idea? Now, as a third output, we have the relative uncertainties, and that will give us even a bit more information about these uncertainties. So, this is color scaled. Let's color scale it between 0 and 100. And now we have a very nice map that provides us loads of information. Interestingly enough, so the vegetated areas, they have very low uncertainties, while this region here has very high uncertainty. So maybe now comes a clue. When we did the field measurements, we were interested in vegetation, and we went only measuring over the vegetated areas, right? We did not do measurements on these bare soils here or so. So that's why that it has these very high uncertainties, because the model has not been trained for it. So basically, it tells us the estimates are maybe good, but it tells us, hey, I'm not so sure about it. You better go back to those regions, get some more samples if you also want to map that with high certainty. So that's a very nice thing of uncertainties. It gives you information where you have to go into the field for doing some additional sampling. And typically, agencies are interested in a map with a certain uncertainty. So we could also apply a mask of our map based on these uncertainties. So let's do that. So we say that we want only to show our estimates when it falls within uncertainty between 0 and, say, 30%. So we develop our mask, and we apply it. So finally, we have a map with outputs where we are certain of that this is produced with enough quality. And we can see basically it provides our estimates of our vegetated areas, and the bare soil or the parts where the model was not sure about, those are masked out. Okay, so this is a first part of what we can do with the toolbox. Now let's have a look to some more options. So I will close this all. And let's go back to settings. So I go back to single output. And maybe I will remove a few of them. Now let's have a look to, since it has 64 bands, or 62, I will apply this dimensionality reduction to see if that actually helps in improving the models. So I go to Band Tools, Dimensionality Reduction. I will select Principal Component Analysis. I could select all of them, but it takes a long time, so I will just select 2 of them, PCA and partially squares. And let's say just 5 components, that's fine. And also because actually we had about 100 data points, so we used 50 for training, 50 for validation. Now let's try to use all the points that they all are being used in validation. So let's use cross-validation, and let's do a k-fold of 3 subsets. So this is deactivated. I click again on Done, give it a new name. And let it run again. So now it will take a little bit more time because we do not only the 3 variables, but we also do for each variable, for each method, 3 subsets with this cross-validation. On the other hand, we also apply 2 dimensionality reductions, PCI and PLS. So it loops also over that, but since it is being reduced, it should go faster. And here are the results. So we can see that for leaf chlorophyll content, the best performing dimensionality reduction method in this case is PCA. So I only show the top best results. If I say top 2, then also the other results will be shown. So Gaussian processes first with PCA, second with PLS, then kernel-rich regression, and then linear regression. And now let's have a look again towards these sigmas plot. Now we no longer see the 62 bands. No, why? Because we first applied this dimensionality reduction. So now the regression is based on these components, and we can see it especially makes use of the first component and the 5th one. Meanwhile, the 3rd one was less important for this regression model. So we again have the... Also, I don't know if the results improved as compared to our 3rd exercise, but very often when you apply dimensionality reduction, especially when you have many, many bands, there's this problem of redundancy that causes trouble. Often when you apply this dimensionality reduction, your results actually improve, so you get more robust models. So this is for leaf chlorophyll content, same for LAI, same for fractional vegetation cover. So let's do again some mapping. We select one. We click on Done. We go to retrieval, image. We select it. We select our image. And we select output. In principle, this should go faster since the data is now being compressed into these components. And again, we have our image. So this is leaf area index. So we have our estimates. And again, we have our uncertainties. So again, we can apply a mask. And let's say that we only want to show our outputs between 0 and 30%. Again, it's more certain over the vegetated areas, meanwhile the bare soil parts have been masked out. Okay, so so far these are single output methods. Now let's have a look towards multi-output methods. And that's really cool because then just with one model, you have produced all kinds of outputs. I will use these 3 variables to create this single model, and then immediately we have maps of these outputs. So I will close this again. And settings, now I go to multi-output. Here we have 5 options. I will not select neural networks because it takes too long training time. So these ones, and let's do it again with some PCA. Okay. Here we give again a name. And let it run again. This again should go faster because only one model is being generated. Here we have the outputs for these different methods, and we can see that in this case, kernel-rich is best performing, but it's very close to support vector regression and then Gaussian process regression. So maybe now let's select this kernel-rich regression for mapping. So we select it, and again, go to retrieval, image, we select our image, and our output. And now what we have as output, we have no longer just LAI or chlorophyll content, but actually we have now immediately 3 maps. So we have our chlorophyll map, this one. We have our LAI map, this is 2. And we have our fractional vegetation cover map. This is 3. So, well, the message is basically that it's very simple to use these methods, and you can apply it to any kind of image, and immediately you get your maps. Okay, so, maybe with this I want to close the first part, so the machine learning part. But now you have understood the principles. Exactly the same idea is done for the vegetation indices. Well, since this was the second most voted topic, I will now go towards vegetation indices, and we do the same kind of exercise, but then with vegetation indices instead. So I will close this toolbox. We go again to Artmo, and now we select spectral indices. The core idea of spectral indices, we will not just calculate an NDVI or what other existing indices. No, what we will do is that we will use the NDVI formulation, and we will calculate all kind of possible band combinations. So we use all possible band combinations for the red band and all band combinations for the near-infrared band, and then it will calculate the best performing one, right? So with CRIS, we have about 62 bands. It will be 62 by 62 combinations. It goes very fast, but you can even develop a 3-band index, 4-band, up to 10 bands. So it will do all the possible combinations of all these bands. So rapidly, you do millions or millions of combinations. In this way, at the end, it will show you the best performing index, so to say. So we go again, loading our data. I can be a bit faster. I had saved it before. So if I click just on load, I just load these settings. Okay, here we are. I click on import again when it's ready with loading, okay. So you can see it's exactly the same way organized, right? We go from input settings, we do the validation, afterwards retrieval. So in settings is slightly more complicated than this machine learning. With machine learning, we just have to select the methods, and it does the calculation for you. Meanwhile, with indices, these are parametric methods, so we need to make a few parameterizations. First of all, we need to select what kind of methods we want, what kind of indices we want to evaluate. We organize them according to broadband indices or narrowband or leaf pigments or water indices, but essentially it just uses a formulation and not the specific bands. You could constrain it to specific bands, but now we keep it open, and so we select the NDVI formulation, and you select a fitting function. In this case, we will just select linear, but we also have exponential, logarithmic, polynomial. And again, we have to say how much data we use for calibration and how much is used for validation. So we click on done, and we say again a new name. Okay, so now it's calculating all possible combinations according to NDVI for these 3 variables. And it shows me the top performing one, and it gives me what are the bands that led to the best results. But this is the top best one. But let me also give maybe the top 10 one. So this was the best performing, this band combination, the second best performing, and so on. Again, we have the same options for plotting our validation data measured against estimated. We can also reorder our index so that the first band is the lowest, and that looks nicer. And also, by the way, it gives you immediately also the equation, right? So that you can see what is actually being done there. And also, you have this measured versus estimated. So it's not as good as the machine learning, but still it's quite okay, I would say so. But maybe more interesting is and I will just activate one option, to show all the potential, or all the possible outcomes. So, and this has been done in the correlation matrix. I click on plot, and now it will give me all the results. And let me show this as a function of R squared. It's easier to interpret. Okay. So here we can see immediately all the potential band combinations, and the more yellow, the better performing. So basically we can see that in the red edge part, which is here, and near infrared, appears to be the strongest region for, in this case, leaf chlorophyll content estimation. It makes sense. Red edge is very sensitive towards variation in chlorophyll content. This region here is between 500 and 600. It's this one. Is a typical PRI region, so it's also a sensitive region. So this is now being done for chlorophyll content, and we can do the same for LAI. So let's select again. It will look pretty similar, I would say so, because also chlorophyll and LAI is kind of related in this data set. When we have high LAI, we typically have high chlorophyll content because it's on these irrigated areas. So you have on one hand dry with low LAI, low chlorophyll content fields, and then you have the irrigated fields which has very green and very high LAI. So we see here indeed that, again, the red edge region together with near infrared is best performing for LAI estimate. Let's do the same for fractional vegetation cover. Basically, it will give the same kind of results. Okay, so once we are happy with the best band combination, we can select it and we can apply it to an image. So I click on done. I go to the next step, image, and I select. You could, again, configure it yourself, but we did already, and it has been validated. So we select this model, and we apply it to our image. We give an output name. You can imagine this goes much faster than machine learning, but we don't have uncertainties anymore. So, and also, this map didn't look so nicely. So this also shows that machine learning typically is much more adaptable or flexible than traditional indices. At the end, we were using just only 2 bands. It was working nice on our validation data, but when you apply to a complete image, you can see the map does not look so nice as the machine learning maps. Okay, with this, I want to close also the vegetation indices. So maybe just for you to know, so basically you can combine any kind of index with any kind of fitting function, and then you can develop your own indices, right? So now I used ones that are already being provided, but within tools you have the options to add spectral index, and there you can define yourself with as many bands as you want, and it will do then all the possible combinations with it. So maybe to finalize, let's try some Sentinel-2, because this is maybe nowadays more challenging and more interesting. And let's do that again for machine learning, because that's more powerful. So I will close. Go to machine learning. So first of all, again, we need to collect our spectral data. Basically, what I did, I used this CRIS data set, and I resampled it towards the Sentinel-2 bands. Because very important is that you always have your machine learning algorithm is exactly the same bands as your image, right? It needs to match. So always take care of that. So we load some, I think I can go to load, to data to Sentinel-2. We have our, in this case, I used 10 Sentinel-2 bands. Basically, I used all the 10 meters and 20 meters bands, and I discarded the atmospheric bands of 60 meter. So we see again our data, and you can see that we have here our 10 bands, and then the spectral data and the variables on top. So I import. And let's immediately select the most powerful one, these Gaussian processes. And maybe we do cross-validation so that all the data is used for validation eventually. So we run it. And then afterwards, we apply it to a Sentinel-2 image. So I will not process a complete Sentinel-2 image. It takes too much time. I just took a random subset of it. Are we happy with the results? Let's have a quick look. For chlorophyll, so-so. LAI maybe is a bit better. LAI looks quite nice, so with R square of 93. So let's take this model. So we select it, and now we apply it to Sentinel-2. So I go now to Sentinel-2. And so I have the full image, and I made a subset of 4,000 by 4,000. Let's select that one. And then the output. OK, so this takes a little bit more time. But it shows that it's working, and you can apply it to your own images. Are there any questions meanwhile? Yes, also a uncertainty map will also be created, yes.
I was asking whether he can create an uncertainty map, but he already said yes.
So uncertainty so far is only with these Bayesian statistics. We have two Bayesian statistics, which is Gaussian processes, and then there is also variational heteroscedastic Gaussian processes. On the other hand, if you do lookup table-based inversion, you could, so it does the inversion against the relative transfer models. You have, it was also explained yesterday, so you have, it does the spectral match. You have your LAI or chlorophyll, but also you have the residuals, how much the difference between your pixel and the simulations. And that residuals, the more it is, the poorer your spectral match. So that also can be interpreted as uncertainty. However, this Bayesian statistics is much more straightforward. So here we have our output map. And maybe it takes a bit more time to open. And this does not look super beautiful, but let us constrain LAI between 0 and 5, maybe. And this looks better. So this is our, I think it's a region over Toulouse. And let's also have a look to the uncertainties now. So the absolute uncertainties. Maybe let's also constrain it between 0 and 3, maybe. So overall, it looks that they are quite low uncertainties, and maybe to close with the relative uncertainties, that gives a bit more information. This does not look good, but let's constrain between 0 and 100%. And now we can see that actually the uncertainties are quite okay. And we can also see that they are, again, low uncertainties over vegetation because, again, it was trained for vegetation, while higher uncertainties are over bare soils or man-made surfaces. So maybe to finalize, let's make a mask. And we constrain between maybe 0 and 50%. So let's apply the mask. And maybe with this, I can close this session for this morning. Oh, let's show it a bit more beautiful. Okay, so this is our final map, and with this, this is a broad overview of the toolbox for today. And tomorrow we will go a bit more into detail about the iterative transfer models, how to use that. It's also very easy. We will, again, step by step, how to configure it, how to apply it, and then show some applications of these RTMs. So if there are any questions.
Thank you very much for the presentation. I think it's a brilliant application. I just wanted to ask you about, well, from the beginning of your presentation, you were saying that with the flex mission, you can detect the stress.
Yes.
And as you say, stress is a very dynamic process. So how can you dissociate occasional stress from a long-term process of decline, vegetation decline?
Yes, so actually stress is very, very, very complex because also flex flies over only one time at a moment during the day. Meanwhile, this dynamic behavior of fluorescence as a response of stress changes over the day. Basically, in the very morning, under normal condition, it's not yet under stress, right? So the first light is used for photosynthesis. But as soon as there's too much light coming, so by noon more or less, then the plant starts to become under stress, and then loads of the light is no longer used for photosynthesis, but rather is being dissipated by means of all kind of photochemistry regulations, MPQ. So that's already a thing that you have to take into account, the time of the day. Then further, flex also is not flying every day, but at best every 20 days or so, depending on where you are. So that's why actually Flex is just an Earth explorer. It's more a technology demonstrator. So maybe for operational use of stress detection, it's not yet ideal. But anyway, when you have a map over a certain region and you see variations, probably these variations are due to differences well, in stress conditions of the plant.
And I think you were saying that the pixel size is 300 meters.
Yes, 300 meters, yeah.
I mean, it's, in my opinion, it's too coarse pixel, you know, it's not possible to make it narrow. You know? No,
indeed, 300 meters, because it's global coverage, of course, right? So I think with 300 meters, 300 meters is fine for more to see how the photosynthesis is at partial scale or landscape scale. Also, 300 meter has been chosen because it flies in tandem with Sentinel-3, which is also 300 meter. So maybe one day there will come a mission that will fly in tandem with Sentinel-2, and then we can go down to 20 meters. But so far, Flex is this Earth, this technology demonstrator. If it appears it works out, let's hope so. There will come a new Flex mission, maybe with a higher spatial resolution.
Okay, any other questions?
When you did the dimensionality reduction and you showed which components were giving the most information, can you then look into that to see what spectral bands are grouped into the different components and then individually choose what bands you want to use for, if you wanted to just say have 5 bands? Yeah,
I will write down on my to-do list. This is still yet something to be done. It's actually interesting indeed and should be possible, but so far we have not implemented such an option. I think if you are interested, we can give you the source code and you could try to find out yourself where exactly this information is stored. But so far it's not yet an operational option.
Anyone else? Yep.
Thanks. Does this like software work just with continuous variables, or can you add categorical variables in as well? Sorry, is that better?
Okay.
Can you add categorical variables in as well as continuous variables when you're using this software?
Yeah, so for us, the variables are just numbers, right, at the end. So anything that you put in Excel, so to say, so that we can convert to a text file, that can read. So in principle, it does not depend what kind of variable it is, as long as it's a continuous variable. So this machine learning, by the way, is for regression, right? It's not for classification. So as long as you have a continuous variable, can be ocean, atmosphere, vegetation, it should work.
Okay, thank you.